Sweet. Got a fair bit to do today. So what I'm going to do today is cover Chapter 17 on the mechanisms of transcription and translation. And I'm going to go pretty quickly because I'm going to follow more or less the book, and I'm just going to try to highlight to you the most important things that I want you to take from it. You should obviously read the chapter, but I'll go through it using the figures from the book for the most part. I'll have some deviations from that. We'll go through that, and it's quite a bit to cover, so like I said, I'll go relatively quickly today. So the central point of understanding transcription and translation is that that's the way that information from DNA manifests itself, right, for the most part. There are some exceptions to this, and we'll talk about them next week. And next week we'll also pick up on the dynamorph stuff that I talked about, and I promised I asked you a question about that. We'll pick that up later. But what we're going to do today is talk about how you go from a DNA sequence to an RNA sequence to a protein sequence, because essentially, right, that's the genetic information is what's in the DNA sequence, and that's what you inherit. But the way that the phenotype ends up being manifested is because that DNA sequence usually is output as a protein sequence, and that is what obviously affects the phenotype. So, for example, when we talk about like the white gene in Drosophila that gives white eyes, so the normal copy of that DNA is translated as an RNA and then made into a protein that is actually as a transporter that allows a precursor molecule to go into the cell that it needs to make the pigment. And so a mutation in that DNA is then a mutation in that RNA, and then that protein is non-functional given a particular mutation. Okay, so a quick overview is that, again, the information content of the DNA is in this nucleotide sequence, and we'll talk a little bit next week that there is some information in DNA that actually exists separate of the nucleotide sequence, and that's a more modern concept known as epigenetics. But and it's that DNA that, of course, is inherited from generation to generation, and it's the proteins for the most part that link genotype to phenotype. Okay, and then there's the concept of gene expression, and we'll talk much more in detail about gene expression on Monday and how that's controlled. But then anyway, that involves then transcribing that DNA and then translating that RNA. Right? There are some exceptions. So the book makes a point in this chapter about how that it's all DNA, RNA, protein. But in fact, there are quite a number of cases where the DNA, the functional part of that, or the functional product is actually an RNA, not a protein. Okay, but we'll focus mostly on the concept of going to protein, which is probably the majority of genes do that. Okay, so RNA is then the bridge between the genes and the proteins that are encoded. And transcription is a process of synthesizing RNA using information from the DNA. Okay, and so transcription from DNA, the product of that is called the messenger RNA. Okay, and then you use that messenger RNA to generate a protein sequence, and that's the process of translation. So you synthesize a polypeptide using the information that's on that mRNA. And ribosomes are the structures that actually are responsible for translation, and they're composed of both protein and RNA, as we'll see later. Okay. So it turns out that in prokaryotes, translation of mRNA can begin even before transcription has finished. So I'm going to show you models of how you transcribe DNA into RNA and how you translate it. And in prokaryotic cells, you can actually start translating it even before you finish transcribing the whole RNA molecule. Okay? And that's because there isn't a nuclear envelope, right, in something like bacteria and archaebacteria. And so all the machinery needed to do both transcription and translation has access to that right against the DNA. And so both of those processes can go on at once. Okay? But in eukaryotic cells, okay, again, one of the defining features of that is having the nuclear envelope, and that actually causes a separation between transcription and translation. So transcription occurs in the nucleus, right? So the DNA stays in the nucleus, so that transcribed messenger RNA is made there, and then that messenger RNA is modified, it's processed, and then it actually exits the nucleus. And the ribosomes, which can do translation, are in the cytoplasm and not in the nucleus. So then translation is restricted to the cytoplasm, and that's just illustrated on this slide. So you can do the transcription reaction off the DNA in the nucleus. You make a messenger RNA, what we call a pre-mRNA, and there's various processing that has to go on in a eukaryotic cell. In a eukaryotic cell, to that messenger RNA to make a mature messenger RNA, and then that actually exits the nucleus, and then only then can it be translated by the ribosome into a growing polypeptide chain. Okay? So the primary transcript is the RNA that's made before processing. Okay? And then again, the central dogma that we're illustrating here is how you go from DNA information to RNA information to protein. Okay? And so we know that proteins are made up of amino acids, and you learned in the first part of the course that there were 20 different amino acids, and you used whatever combinations of those you can and whatever strings, that's what's used to make a protein. Okay? And the protein can be like a structural protein that, like a keratin that makes your hair. It could be an enzyme that catalyzes some reaction. It can be any of a variety of types of proteins. Okay? But they're all made from these 20 amino acids, right? And so one of the questions is, so what is it really in the DNA that's the instructions for the protein sequence? And in DNA, there's only 4 nucleotide bases, right? A, T, C, G. And so how can you turn that information into protein information? And so, right away, people thought, well, okay, you must be able to read along it. And if you think about it, how could you translate that eventually into a protein sequence? And so if you could get information from a single nucleotide position, that would only give you 4 possible things to code. If you get it from 2, right, that would give you 16. Okay, and so you don't get up to 20 or greater, right? Obviously, when you get to 3. So that led to the idea that there was this part of the idea that there was this triplet codon, and that's going to be one of the central themes is that we're going to read this as triplets, okay, of nucleotide pairs. Okay, so the flow of information from gene to protein is based on a triplet code. Okay, it's a series of non-overlapping 3 nucleotide words. That's how you're going to read the DNA. Okay, and so the words of a gene are transcribed onto a complementary non-overlapping 3 nucleotide word that makes the mRNA. So we talked about DNA replication, how you can faithfully copy a molecule of DNA to another molecule of DNA. But in this case now, for transcription, we're going to copy the DNA as RNA instead. Okay, and that's going to be our messenger RNA. And then we're going to be able to take that messenger RNA and eventually use that information to synthesize the correct polypeptide chain. Okay, so this is the basics then of that flow of information. So you have a double-stranded DNA molecule, and again, it's got these base pairs in it, right? And in transcription, you're going to separate these strands, and you're going to use one of them as a template, and you're going to end up making an mRNA strand. Okay, so a copy, but instead of being made of DNA, it's made of RNA. Okay, and then you can read these as triplets of 3, and we'll describe that in a little bit. And then that's used to determine what the amino acid sequence is. Okay? And again, for those of you who've taken the lab or taking the lab now, it's very familiar to you already. Okay, so during transcription, one of the 2 DNA strands is used as the template. It's the one that's red. Okay, and it's red in the sense that then you synthesize an RNA strand that matches that's complementary. Okay, and for a given gene, the template strand is always the same strand, and we'll explain in a second how it is that you end up that way. Okay, and then again, in the subsequent process of translation, that mRNA is read in triplets. Okay, that are called codons. So those sets of 3 are known as codons. And the other thing is that like many processes that involve nucleic acids, everything goes from the 5' to the 3' end. So when you make the messenger RNA, it's synthesized 5' to 3'. Okay, so it uses the template strand that's the other direction. It's 3' to 5'. Okay, and then also when the messenger RNA is read as triplets, it's always read in the 5' to 3' direction. Okay, and it's going to be read that way so that the 5' end of the RNA is making the C terminal part of the protein, and then the 3' end is making the N terminal part of the protein. Okay, so it's a very orderly linear arrangement, all of this information. Okay. So codons are read, again, in the 5' to 3' direction, and each codon specifies an amino acid. Okay, and there's 20 amino acids that you're trying to do, that you can specify along this polypeptide chain. Okay, and it turned out that people figured out what the code was, and that was actually a major breakthrough. So shortly after the structure of DNA was discovered, a number of people then were interested in trying to understand how that was read. And they realized that you made RNA from it, but then they didn't know how you actually read that as protein. And so in fact, Francis Crick, who was one of the co-discoverers of the structure of DNA, that was one of the things he was also very interested in, is understanding the genetic code. And the first breakthrough came by just being able to synthesize long strands of RNA that were the exact same nucleotide over and over again. So for example, to make a strand of RNA that was just used over and over again. And then actually to get that translated into a protein by the machinery in the cell. And what you found is that made a particular polypeptide of just a particular amino acid over and over again. Okay, so then you knew what U encoded. And you could do the same thing for C and G and so on, right? And then people could get fancier and synthesize particular triplet repeats and then understand what the genetic code was. So in the end, that it was deciphered, and was found of the 64 possible codons then, right? So 4 possible positions at each of the 3 nucleotides. There's already 64 triplets. So 61 of them code for amino acids, and we'll show you the chart in a second. And 3 of the triplets encode what's called a stop. So it says now, okay, now end, and don't translate anymore. And then there's a specific one that encodes an amino acid. It's also the start. It's always where you start. Okay. And the genetic code then is redundant. So there's 64 triplet codons. There's only 20 amino acids. That means there's some redundancy to the code. And usually it's that third base pair of the triplet that is what's called a wobble position. So there's a variety of amino acids. There's a variety of triplets that can be used that still encode the same amino acid. So that there's that redundancy. Okay. But it's not ambiguous. So a triplet encodes a very particular amino acid. Okay. And the codons must be read in the correct frame, too. So it's a series of triplets, and it's read as triplets the whole way along. And so as we'll show you in a minute, if you insert a single nucleotide into that region, it's going to throw the whole frame off, and you're going to get the wrong thing after that insertion, okay, or a deletion. So if you insert or delete 1 or 2 nucleotides, that's going to shift the frame. If you insert or delete 3 nucleotides, or some multiple of 3, then you're going to just insert or delete a couple of amino acids, but the frame will stay the same. Okay, so this is the codon table. This is sort of the standard codon table that's been discovered. And it's actually a fairly universal codon table. There's only a few organisms that have small violations of this genetic code. So it's a very ancient genetic code. And so it's in this particular table, it's written as the triplets that are in the RNA. So instead of T's, you're seeing U's in these positions, okay, or T's in the DNA. And you can see then that there's a variety of ways to encode different amino acids. The 3 stops are these. The start is this AUG, which is always at the start of a particular polypeptide, and that ends up being incorporating methionine then at the start of a protein chain. So you can see the levels of redundancy. So certain amino acids, right, like arginine, you'll see that there's actually 6 ways to encode them. So there's this third position that can be any of the nucleotides, okay, and that's still arginine, and then there's this other additional complexity where the first, a first nucleotide can be different, okay. A lot of them there's just 4 ways to encode them. A few there's 2, and in a few instances like methionine there's just the 1 way, okay. So that's the code. Okay. And this code is nearly universal, okay? And it means then that you can take a gene from, say, bacteria, and you can put it into humans, and it's translated as the same polypeptide chain, okay? And that's because the code for reading it is universal. So the machinery to make the RNA is obviously just making a copy, but the code that's in the cytoplasm involves the ribosome is the same in all of these cells, okay? There can be a difference in efficiency, and we'll get to why that is a little bit later. So there's particular reasons why they could be more efficiently translated in bacteria than in humans, and that has to do with some biases in the usage of particular triplets, okay? So again, transcription is the first stage of controlling gene expression, okay? And so this RNA synthesis that you're making off the template DNA strand is catalyzed by an enzyme known as RNA polymerase, okay? And again, RNA polymerase is actually several proteins put together into a complex that's able to accomplish this, okay? And it actually pries apart the DNA strands, and then it's hooking together the RNA nucleotides that are lining up to that template strand, making then a string of covalently linked RNA nucleotides, okay? And it's complementary then to the DNA template that it's copying or that it's reading from, okay? And the RNA synthesis follows the same base pairing rules. Again, the only difference is that anytime you think of having a thymine in the DNA sequence, you're using a uracil for RNA. Okay, so then there's a couple of things that it needs, and we'll illustrate a very simple set of this with bacteria first, and then on Monday we'll talk about the more complicated things that go on in eukaryotic cells to initiate transcription. Okay, and so there's a stretch of sequence called a promoter, and this signals where transcription is going to start. It starts just a few nucleotides downstream in the 3' direction from the promoter. Okay, and then in bacteria there's a sequence called the terminator, which tells the RNA polymerase to stop. Okay, so there's a start and a stop to this transcription process, and that transcribed unit, we call it the transcription unit. Okay, so here's kind of an illustration then of the process. So RNA polymerase will sit down, it will open up the strand of DNA, the double helix, and then it'll start to synthesize then an RNA transcript. Okay, and again, the polymerase is using this, the promoter is going to be using sequence it's reading is on this strand here, and it's read in the 5' to 3' direction, but then it's synthesizing RNA then in the 5' to 3' direction as the polymerase goes along the DNA. Okay, and it can go, I think the book is something like 40 nucleotides a second, so it chugs along. It's not as fast as DNA polymerase, but it moves along and it's making this transcript. And then again, when it hits the sequence called the termination sequence, the RNA polymerase will fall off and release this completed RNA transcript. Okay. And so that process is called elongation. So that's where it's elongating the RNA, the synthesized RNA molecule that it's making. But there are 2 other steps we'll need to tell you, and that's how it initiates and a little bit about how it terminates. So it's chugging along, but how does it know, again, where to start and where to stop? Because that's important, especially that it know where to start. Okay. And so we have these sort of minimal DNA elements we call promoters, and they signal the start point, okay? And that the actual start point is a few dozen nucleotide pairs downstream from where that actual promoter sequence is, okay? So that promoter sequence gets the polymerase ready to go, right? And then it starts. And then there's a series of things called transcription factors, and we'll go into more detail about them on Monday, that help to mediate the binding of the polymerase to that transcript initiation point, okay? And that can then control when genes are on and off those transcription factors. So we'll talk about examples. For example, there's an enzyme that bacteria make that let them use lactose as a carbohydrate source, but they don't need to make it if they have other sources of carbon around, and so that can be regulated, and the transcription factors will regulate whether transcription really gets started or not off of a promoter site. And like I said, we'll talk more about that on Monday. Okay. So the completed assembly of the transcription factors in the RNA polymerase, which is called RNA polymerase II, that's called a transcription initiation complex. So you have this whole complex of proteins that's sitting there ready to start transcribing. And the promoter is called a TATA box, and you'll see why in a second. It has to do with the actual sequence. Okay? And it's crucial to forming that initiation complex in eukaryotes. But there are exceptions, especially when we get into thinking about prokaryotic gene regulation. It's a little harder to recognize the promoter just by its sequence. Okay? So you have, the reason it's called the TATA box is you can see that in the sequence. It's something like TAT and then a string of A's. That signals this promoter sequence. Okay? And so what's going to happen is there are other factors. There's usually other sequences in this DNA that initiate the binding of other specific proteins that vary from gene to gene, though. And then that sits down. It recruits helps to then stabilize RNA polymerase II. Okay, and that then is the site where then transcription is going to get started. So it's going to start just a little bit downstream from that actual TATA sequence. Okay, and it's going to start going this way. And so this is why there's a specific directionality to it, because you read the TATA sequence on one strand. Okay, and then you're going to go on that strand 5' to 3'. Okay, this direction is the direction the polymerase is going to go, and that allows you to make the complementary strand. And then since all of these polymerases can only themselves catalyze a reaction or catalyze a polymerization of the new strand in the 5' to 3' direction, so it's having to go this way. It's a lot like a DNA polymerase. If you think about the topology of it, it's having to go only one direction. It can't go in the other direction. Because of where it starts. So it sits here, but then its ability to polymerize actually starts right there. So it doesn't start right at the TATA. Okay. All right, so as it's moving down, it has to also untwist the double helix, and it can do that. And it does it sort of in a 10 to 20 bases at a time. And like I said, it can go about 40 nucleotides per second in bacteria. Okay. And it turns out that several POL2 enzymes, RNA polymerase II enzymes, can hop on at the same time, sort of in a train, one after the other. And so you can actually be synthesizing a lot of RNA molecules off the template simultaneously, right? So the different polymerase molecules will have gotten different distances down the transcription unit, and they'll all be making new mRNA chains as they go. Okay. And so the nucleotides are always added to the 3' end, because again, all of these things always make these things are always made in the 5', 3' direction. Okay. And so that just shows the process in a little bit more detail. So it's unwinding the DNA and then splitting the strands, and then it's synthesizing the new RNA molecule. So it's moving this way along the DNA, and then it's synthesizing a newly made messenger RNA molecule. Okay. All right. So then it also has to terminate somewhere, and the mechanisms of termination are different in bacteria and in eukaryotes. So in bacteria, the polymerase stops transcription at the end of a sequence called the terminator, and then the mRNA can be translated without further modification. Yeah. What's that? Right. So the sequence is, if you look at this, right, the sequence is not the same on the two sides, right? So you can only read this as the TATA box, all right? So it's going to give it a direction. Does that make sense? Okay. Because on the other strand, right, it doesn't read that sequence. It's headed in the other direction, but it's not a TATA box. It's not that sequence. Okay. So it's going to head in this direction. Okay. It turns out that the reality is in bacteria it's pretty simple. In prokaryotic cells, like in human cells, it gets very complicated as to what the promoter really is. Okay. But we show you the bacteria is fairly easy to understand. Okay. In eukaryotes, the RNA polymerase transcribes what's called a polyadenylation signal sequence. So there's actually a sequence that it runs into that we'll describe in a minute, or tell you the significance of. It's going to actually cause a string of A's to be added to the RNA later. Okay? And then once it reads that sequence, the RNA transcript is released anywhere from 10 to 35 nucleotides past this polyadenylation sequence. Okay? So that's what goes on in our cells, for example. So it's a little bit more ambiguous because the sequence for polyadenylation is a little bit harder to characterize. And so in some genes it's pretty obvious, in others it's not. We only know it because we know that we can sequence the mRNA separately and know exactly where it was terminating. Okay. So but in eukaryotic cells, there are various steps that still have to go on to turn that messenger RNA, called a pre-mRNA, okay, into a transcript, into an RNA that can really be translated, can leave the nucleus and be translated. And both ends of the RNA get modified, okay. And the curious thing in eukaryotic cells is that the interior parts of the RNA molecule get cut out, okay. We'll show you in a second. These are called introns. And then the remaining pieces are spliced together, okay, to make the mature RNA. Okay. So two of the things that go on are additions of sequence to the two ends in eukaryotic cells. So the 5' end receives what's called the 5' cap. It's a string of modified nucleotides. It's very specific. They get stuck on there. And the 3' end gets a series of A's added to it. Okay? And these sequences, part of their function is they give stability to the RNA. So the RNA goes into the cytoplasm. If it were to be missing these sequences at the end, they're enzymes that would hydrolyze the RNA and destroy it. So these provide stability. And then the cap at the 5' end is also important for the ribosome to get started, to turn it into protein, to read it out as protein. Okay? And they also facilitate the export of this mature RNA from the nucleus into the cytoplasm. Okay? So this is just sort of a summary diagram then. So you'll have this transcribed region that was made off the template of DNA, but then these two yellow regions are added after it's been transcribed off the DNA, but before it leaves the nucleus. Okay? The cap and the poly A tail. All right? And you'll have, within this message, if you read it, you'd be able to see a position for a start codon, the AUG, and then one of the 3 stop codons. Okay? And we'll talk about how you know where those are. Okay, so, but it turns out again that in many eukaryotic genes, there are long non-coding regions of nucleotides that sit between the coding regions within an individual gene. Okay, and these are called introns. And their discovery was very surprising. No one had any idea that you would have this RNA, and then you'd still have to actually process it to get the right sequence out by cutting regions of it out. Okay, and these are called introns. And then the parts that are left that are actually translated are called exons. Okay, and they're the ones, and then what's left then is translated into amino acids. And it's a process of what we call RNA splicing that removes those introns. So that's the process that's going to cut out what are the intervening sequences. Okay, so it in principle sort of looks like this. So again, we have our region that was transcribed in the cap and the poly A tail. But if you look at it, what's going to happen is certain parts of the messenger RNA are actually going to be cut out, spliced out. Okay, and that's going to occur. And then the